So DNA is wrapped around these histone proteins, and then it's packaged up into the chromosome structure. We're going to come back to these special histone proteins because they play a very important role in epigenetic regulation of transcription. So we're going to look at these in more depth in the future. But this is just an introduction, and for you to keep in mind that the DNA is not really naked in the cell. It's packaged up in association, tightly associated with proteins. And there's a lot of it, right? And so there's a lot of DNA. And our next topic is replicating that DNA. So putting aside the histone proteins and back to that helix itself, how do we replicate DNA molecule? Let's just take an overview first of the process. Think about when this would occur. When do we synthesize DNA? So it occurs during the S phase, right? The S phase of interphase in either mitosis or meiosis. So it's when a cell, so before a cell divides. So before a cell can divide, it's got to make new copies of the DNA. And there's a lot of it to replicate, even in a prokaryotic, 4.5 million base pairs, that's a lot of DNA to make new copies of. Human genome, 3 billion base pairs that all have to be replicated with really high fidelity. So we're going to take some time and talk about the process. And, you know, if we go back, let me just kind of, if we think about the central dogma of molecular biology, of genetics, DNA is the starting point, right? It's the recipe. So DNA serves as a template to make new copies of itself, and that's replication. And that's what we're going to talk about here. DNA, before it can actually make those recipes, you have to pull the recipe out into the form of messenger RNA, and that is the process of transcription. And then this information has to be translated into the action molecule, into the protein. Now we're going to cover each of these processes, and each of these processes has a beginning, a middle, and an end. And so there's going to be an initiation process for all of those, and we're going to look at proteins and enzymes that are involved in the initiation of each of these processes. We're going to look at the mechanism of how the synthesis actually occurs, because each of them, you're really synthesizing a new molecule. Here, you're synthesizing new DNA. Here, you're synthesizing messenger RNA. And here, you're synthesizing a polypeptide, which folds into a functional protein. So we're going to do that in initiation, synthesis, and then termination. We're going to look at each of those, or we're going to tackle each of those processes by looking at the initiation. How is the process initiated? How does the synthesis of the molecule actually occur? And finally, how does the process stop, or termination? And I will say up front that a lot of times students conflate these processes, get them mixed up. And so I really want you to, as we go into these, to think about each process in these 3 stages of each process and compare and contrast so that they're distinct in your mind, so that you don't get them mixed up with each other. So back to replication. So it occurs during the S phase, and that's before a cell divides. You have to make 2 new copies of the DNA molecule. The replication process itself is called semi-conservative. So it's a semi-conservative process. I don't think I'll go into the experiment that demonstrated it was semi-conservative, but I will say the scientists, and you'll sometimes hear these, were Messelson and Stahl, who determined that it was a semi-conservative replication process. And so what does semi-conservative mean? If we look here, here are different ways DNA could divide. Here's your parent DNA molecule shown in purple. So in a conservative model, the DNA would serve as a template, the parent strands would serve as a template, but then they would reform, and then the new strands would form together. That would be a conservative model. You could have a dispersive model where you sort of get this mixture of both parental and newly synthesized strands in each in the daughter molecules of DNA. That would be the dispersive model. But what Messelson and Stahl discovered, like I said, we won't do the actual experiment, but what they discovered is that it's a semi-conservative model where the parent DNA strands separate, exposing the bases on each strand. So they separate during replication, and each parent strand serves as a template to create a new complementary strand. So the daughter molecules, so here's our parent DNA molecule, results in two daughters. And there's one of the strands of the daughter molecule are old parental strands, and then in blue here is showing the newly synthesized strands. It's semi-conservative. So let's take before we get into all the details and all the weeds, because we're going to have a lot of new proteins, DNA sequences to deal with. So before we do that, let's go ahead and just do a mock sort of synthesis. Now, synthesis always occurs in a 5' to 3' direction. And we have to remember as well that the base pairing that occurs is always anti-parallel. So let's have a mock piece of DNA. We'll put in a little sequence here. Here's the complementary strand. So it's going to be CTAA-TGT. Let's make some directionality here. So this is our parent molecule. So what happens is that the strands are going to separate. And the new strand is going to be synthesized in a 5' to 3' direction. So here's the parent strand serving as a template. Here's our new strand coming in, TG, because this is the 5' end, because we have to be anti-parallel to have these base pairs form. So it's going to go in this direction, TAA, TC. And it would continue in a 5' to 3' direction. On the bottom strand, synthesis occurs in a 5' to 3' direction. So that's just sort of an overview, and we're going to get into like I said, the details and the mechanism next. But always remember 5' to 3', 5' to 3'. That's the only way the chemistry occurs. So let's start with initiation in the molecular mechanism. Of replication. So the initiation process, and I would say for each replication transcription transcription and translation, the initiation process is the most complex. It has the most things going on to consider. So let's talk about the initiation of replication. The location for the initiation of replication, it's not a random situation. DNA molecules have specific sites where replication occurs, and it occurs at sites called origins of replication. So origins of replication. And in prokaryotes, and we'll look at the differences between prokaryotes and eukaryotes throughout. In prokaryotes, where most of this stuff was originally identified and named and characterized, they have one origin of replication. Remember, they have a circular DNA. So here's the origin depicted here in yellow. Here's the origin. In E. coli, it has a specific name. It is called ORI-C. So the origin in E. coli is named ORI-C. And what we'll see is what is located in this origin. The thing that makes this origin is that it is rich in, it has AT-rich regions. And DNA A boxes. So let's take a look at the origin ORI-C in E. coli. Sorry about that. I was missing an image that I wanted to show you all here. And so here are the AT-rich regions in ORI-C. So the E. coli ORI-C, notice that it's 275 base pairs. So as I said, this is not something that is random. This is the sequence of the E. coli ORI-C. The AT-rich regions, so notice A's and T's are overrepresented, and then the sequences of the DNA A boxes. So this is kind of a confusing name, but this is a specific sequence called a DNA A box. And this is found in the origin of replication in E. coli, this specific sequence, this 275 base pairs. Now, eukaryotes, remember, have linear chromosomes. And so in eukaryotes, they have many origins. There are many origins on each chromosome. Now, the identity, the sequence of each origin are more complex, as you can imagine, than it is in the prokaryotes and E. coli. But one thing that isn't, what they do have in common with the E. coli is that they're AT-rich regions. So the sequences, it's more complex and more variable than in eukaryotes, but often they're rich in ATs. Now, remember back to the structure of the helix. What do you think is special about AT-rich regions? Remember, the association between the A and T base pair is weaker than it is between the G and C base pair. So if you can imagine in this little yellow region, if there's lots of A's and T's, that helix is a little less robust. It's not, it doesn't have the same sort of force of attraction as it would have if there are plenty of G's and C's. Okay, so these are the things, back to the E. coli origin. So here's the sequence. This is where initiation of replication takes place in prokaryotes and in eukaryotes, also at origins. So what happens here? Why is this the place? For replication to begin, DNA binding proteins have to bind at the origin of replication. And so, I'll do that here. So special proteins called origination factors and in E. coli, the specific origination factor has a name. It's called DNA-A. Remember the DNA-A boxes? That's what's here in yellow. This is the protein DNA-A. So the box is the DNA sequence to which the protein DNA-A binds. So origination factors in E. coli, it's DNA-A, are proteins that bind, that recognize and bind to the origins. And it causes the helix, the binding of these proteins at the origin, pops open the helix. And in blue, what do you think these are? Those are those AT-rich regions, which are weakly associated. And you can kind of just, I really like this image because it shows you, and hopefully you can imagine, remember the DNA is a flexible molecule. Here these proteins bind and they cause this sort of contortion of the helix, which pops open the AT-rich region. So origination factors are proteins that bind to the origin and cause the helix to open. Well, let's say, be more specific, the helix strands to separate, forming what is called a replication bubble. So this is the first thing that has to happen. The origination factors, in E. coli, it's DNA-A, binds to the origin, causes it to pop open, forming this replication bubble. So here's our bubble. Now notice that it's a bubble, and so there are actually two regions where the single-stranded DNA and the double-stranded DNA are present. Replication is going to occur in both directions, and where it's occurring is called the replication fork. I think I've got an easier picture to look at on this next one. Here we go. So the replication fork, so where the single-stranded DNA and the helix, double-stranded helix, these are standard shorthands for single-stranded DNA, double-stranded DNA. So where they meet are called the replication forks. This is bidirectional, so these are referred to as bidirectional forks that open in opposite directions. Now, single-stranded DNA. So before the new strands are synthesized, here's our replication bubble. This is single-stranded DNA before the synthesis occurs. Now, single-stranded DNA is vulnerable to degradation by DNA digesting enzymes, DNases, nucleases. It also can fold upon itself. Like if you had a T here and you had a T A here, if this is a movable molecule, and these could fold on to each other, causing secondary structure to form, or folding or internal folding. And that's not good because we want to be able to see these so that we can synthesize a new strand, right? So there's additional proteins that come in called single-stranded binding proteins. Or SSBs come in and they bind and kind of coat the single strands until the new stuff is created. So they bind to the single strands and they coat it and protect it from internal folding. So they bind to the single-stranded DNA and prevent internal folding and degradation. And another consideration sort of for folding and twisting and turning, and the fact that these molecules are mobile, as this opens, as this fork opens, remember this is a helix. It's a helical molecule. And as you open these single strands, it causes the helix to sort of tighten. And if you can imagine, if this continues to open and open, that it can cause a process called supercoiling. I'm sorry, that's a horrible looking picture. It can cause the helix to coil up on itself as this opens. So there's got to be a way to alleviate that. And I forgot to mention as well, so let's go ahead and put that in here. There's an enzyme that continues this opening process. It's not spontaneous. There's actually an enzyme here that continues to open this. And that is, and it's at both forks, that enzyme is helicase. So helicase is an enzyme that binds to the fork and opens the helix. Separates the strands of the helix. So the initial ones that pop it open are the origination factors, but then helicase binds to the forks and it continues to open it as the synthesis process occurs. But as that synthesis process is occurring, it causes the supercoiling of the DNA helix downstream of the fork. So there's an additional enzyme that's going to come into play down here at the fork, and this is going to alleviate the supercoiling, and that's called topoisomerase. This figure is getting awfully messy. Let me see here. So we've got our bubble downstream. We're going to get supercoiling occurring of the helix. It looks awful. So topoisomerase is going to come in. It's going to cut the helix. So enzymes that break the phosphate backbone of the DNA or of any nucleotide strand are nucleases. So it's got nuclease activity, which is going to cut. So topoisomerase is going to cut the DNA using nuclease activity. And it's going to allow the supercoils to unwind. Now, for most of these processes, I've tried to embed a lot of animations because it's hard to visualize moving processes in a two-dimensional drawing. So let's take a look at an animation for supercoiling.
Supercoils can be positive, coiling in one direction, or negative, coiling in the other. Positive supercoiling results from overwinding, like this rope. Negative supercoiling is the opposite. If you unwind a rope and then release the tension on it, it will supercoil in the opposite direction, creating negative supercoils. Let's consider what happens as DNA unwinds during replication. As DNA unwinds, it acts like this rope when we pull apart its two strands. As you pull the strands apart, twisting tension builds up in the rest of the coiled portion. It is actually adding one twist to the remaining rope for each twist pulled out of it. At some point, you can't separate the strands anymore. The remaining rope is too tightly twisted. If you relax your tension on the rope, it will twist around itself in a supercoil. It is releasing tension. If you want to keep pulling the rope apart, you have to release the tension periodically. And one way to do this is to cut the rope and splice it back together. This problem has been best characterized in small circular DNAs. There are two methods of dealing with this problem in DNA. One cuts only one strand of the DNA double helix, and the other cuts both strands. Let's look at the first. Topoisomerase 1 enzymes cut a single strand of the double helix, pass the other strand through the cut, and reseal the break, relaxing the overwound molecule, which now has one fewer twist. Topoisomerase 2 enzymes do the same thing, but with both strands of the double helix. Topoisomerase 2 cuts both strands of a double-stranded DNA and passes another double strand through the break and then reseals the break. So if a molecule of DNA is supercoiled, topoisomerase 2 can remove the supercoiling, 2 twists at a time, to yield a relaxed circle. Supercoils can be positive, coiling in one direction.
So now we have all the players in place for the initiation of replication. Telomerase, single-stranded binding proteins. We have helicase here at the fork. The origination factors that originally bound to the origin to pop it open will dissociate. And then we're going to enter the synthesis portion in the next recording.